Alright, if you watched the first video, you remember that we learned that you have DNA here, and on one end you'll have a promoter, which is usually in the form, at least in a bacteria, as a TATA box. And we learned that there is on the other end of the gene a terminator, and we've discussed this before about exactly what a terminator is. And so in between here, this region on the DNA, this is on a chromosome in a eukaryote, or the little loop of DNA in a bacteria. Alright, so this is your DNA strand. Alright, so it would be double-stranded, so realize that. That's how the loop forms. But what is attracted here, of course, is your RNA polymerase, like in the little film. It makes RNA polymerase. And it's going to attach right here, and it's going to move across this region. Oops, I'm having a hard time here. Yeah, it doesn't want to write. But it's going to move across this region, and it's going to essentially create down here at the end, as it moves across, it's going to create an RNA. Well, one thing we have to understand with prokaryotic genes, that means like bacteria, they operate different than eukaryotic genes. Prokaryotic genes essentially have multiple protein coding regions between the TATA and the terminator. So this piece of DNA that we have here between the promoter and the terminator being, oops, just run out of space there. The terminator being this hairpin loop, and the promoter being the TATA box. In between there, on an operon, this is what makes an operon, you have several regions that are coded for proteins. And in the LAC operon, this is the one that we're talking about. So the LAC stands for lactose operon. You're going to have three specific protein coding regions. One designated Z, the other one designated Y, and finally one designated A. And each of these is designed to produce a specific protein. Alright, so three separate proteins that have three separate functions. This type of gene, an operon in particular, is called a polycystonic gene. Alright, our polycystonic region. And so that means it's a cystron is one of these regions that codes for a protein. So poly meaning many, many regions that code for many proteins, in between your terminator and your promoter. So this is a polycystonic gene, and we need to know what these three do. So Z codes for a protein called beta-galactosidase. I will never ask you that. I mean, you might want to know it, but I will just call that the Z protein. All right, and this one, of course, will be the Y protein, and this one will be the A protein. All right, these are the genes. All right, or the protein coding regions. We can call them open reading frames. There are several terms we can use to describe these, but we'll just say this is a polycystonic gene. Cystonic, yes. And meaning it has several of these protein coding regions that will actually code for a functional protein. Now, the Z protein, it has a function. It digests lactose into glucose. So let me go ahead and start a new picture here. All right. And this new picture, we are going to talk about how a bacteria operates. So here's the outer membrane of the bacteria, and we have some DNA in here, and we have some ribosomes kind of just floating around. Not much to a bacteria. They don't have a lot of parts. They just have some DNA and some ribosomes inside. But let's say there is a gene here. Somewhere there's a promoter and a terminator, a TATA box on one end and a terminator on the other. And that's going to be our lac operon gene. And so it's going to have this structure we were talking about. Very difficult to draw on this. All right, and that is the Z, the Y, and the A. And this one codes for a particular protein, the Z protein. Now, what does the Z protein do? Z breaks down lactose, which is a sugar in milk. All right, lactose. It breaks down lactose into two other sugars, glucose and maltose. So it's a more complex sugar, and the idea is you have to have this particular enzyme to be able to turn it into simpler sugars of glucose and maltose. Now, glucose is the one you want. All living things can use glucose. So all living things utilize glucose and turn it into ATP. Now, if you remember on the first day of class, I said ATP was to your cells like gasoline is to a car. Because what happens most of the time when your proteins want to do some work, they want to change shape. All right, so you have a protein that looks like this and you want to change its shape to do some work or something. It changes its shape to this. Well, to do that, you have to add some ATP. ATP is what powers it. Okay, so ATP is going to power the shape change of your protein, and that's the work that's done in your cells. It's the thing that makes your cells operate, changes the chemistry of these proteins. So by adding some ATP nearby one of these proteins, you have essentially changed the chemistry of the protein. When you change the chemistry of the protein, you change the chemistry of the cell, and you can do some work as well. Well, that's cool. ATP can be used for that, and it looks like the Z protein can be used to turn a milk sugar into at least part of it. It cuts it up and turns it into glucose, so you can make ATP. So this is good for the cell if lactose is present. Now, this is actually some very expensive proteins to make. So the bacteria really doesn't want to make these all the time. So keep that in mind as we go through this, that the bacteria would rather not make these unless it was around milk. And that's the true function of the lac operon. The lac operon is to only turn on this gene when there's lactose in the environment. In other words, if you're not living inside a glass of milk, you probably want to keep this gene, this operon, off. You only want to turn it on when there's milk around, or lactose. Okay, well, we know what Z does. What about this Y and A? They make proteins as well. Well, the Y protein is actually what we call a transporter, and it embeds itself when it's made into the membrane. And what it does is it forms a pore, and that pore is especially designed to let in lactose. All right, so lactose can get in if this gene is, if this operon, I should say, is turned on. You're going to make a protein that can digest lactose and turn it into something useful. You have a protein also that's going to let the lactose in. What does A do? Well, A detoxifies things that accidentally get into the cell, all right, when you make the pour. So this pour tries to be specific and only let in lactose. But in nature, as you might guess, it can't be perfect. There are other chemicals that can sneak into the cell when you make this pour. Well, in case that happens, if those chemicals are present in the milk, then you have to have something that can detoxify the things that sneak in, the poisons, if you want to think of it that way, that sneak in when you make these pours. So A is a detoxification protein. All right, it goes and destroys molecules that usually sneak in when you make Y. So you let in the Z. Z is for the digestion. All right, Y is for letting in the lactose, and A is for detoxifying the things that sneak in. You'll notice that all three of these need to be made at the same time. And that's why they have evolved into this operon. They are all regulated together in this same small unit that we call an operon. They're very close together, and by putting the TATA box, the promoter, up here in front of them, you can make them all at the same time, and then when you don't need them, you can turn them all off at the same time. So that's the kind of the function of the operon. So this is the beginning. This is like a second video here. This is explaining the function and the basic structure of an operon. It's not a normal gene. It has some structures that are a little different.